2, the viral life cycle. Unfortunately, you've been dropped right in the middle of an epic battle, good versus evil. So as the battle unfolds, let's highlight these stages of the viral life cycle and illustrate how alphavir and the rest of the antiretrovirals defend against HIV at every turn. First, the history of this conflict. HIV targets host CD4-positive immune cells, including macrophages and helper T cells, which are HIV's main target. Like other viruses, HIV takes over, i.e. commandeers, the cells it infects, turning them into virus factories. Just as our evil retroviral wizard has seized the neighboring CD4 castle. From that neighboring castle, he's prepared a new offensive, generating new virions, complete mature viruses, that are ready to invade the next cell. Or in this case, new ships ready to invade the next castle. Before we jump into the life cycle of HIV, let's introduce 3 important structural genes: GAG, ENV, and POL. These genes are responsible for encoding the components of the virion and important viral enzymes. As this brand new invading virion ship heads for alphavir's castle, notice it has prisoners on board in the center of the ship, gagged and tied around a 24-hour sundial. This is because the GAG structural gene encodes virion core proteins, like P17, the matrix protein, and P24, the capsid protein. You may recall from HIV Part 1 that P24 is detected through 4th-generation diagnostic testing. That envoy boat is carrying plans for a major attack called the Big One, with siege towers and grappling hooks galore. Envoy also represents ENV, a polyprotein containing the precursor envelope protein GP160, which is cleaved by the host cell into GP120 and GP41. Some quick math suggests that 120 plus 41 equals 161. So, come on, they should have just called it GP161, but that's science for ya. These proteins were represented by the evil wizard's pipe in Part 1, but now he's upgraded from tobacco to war machinery so we can see how they actually function. GP120 and GP41 play an important role in cell entry, which we'll talk about in a bit. Enter Paul the Orkin Blacksmith, a blacksmith of enzymes. He's here representing the Paul gene. Like ENV, Paul encodes a polyprotein which is cleaved to form important enzymes required for viral replication, including reverse transcriptase spellbooks, integrase keys, and protease blades. We'll come back to these enzymes in a bit. Now that you're familiar with the 3 structural genes and the protein products they encode for, let's see how they each play into HIV's life cycle. The outer wall of the castle will represent our defending host cell membrane, and it looks like this battle is about to heat up. Entry into a host cell is mediated by the HIV's envelope glycoproteins. This starts with the attachment of GP120 to receptors on the host cell surface. GP120 binds to both the primary receptor, CD4, and the chemokine coreceptor, CCR5 or CXCR4. Up there on the castle wall, our brave helper T-Squire is under attack. That evil-looking siege tower is binding to the castle wall and preparing to unload its contents. While the GP120 siege tower is for HIV attachment, the GP41 grappling hook is for HIV fusion. Once GP120 binds to the host receptors, GP41 undergoes a conformational change that leads to fusion of the viral membrane, aka envelope, with the host cell membrane. Now we see the orcs storming the castle trying to fuse to the wall with these GP41 hooks. Once HIV enters the host cell, the virus uncoats, releasing viral RNA, enzymes, and structural proteins into the cytoplasm. And once the wizard gains entry into the castle, he gets to work, turning the castle into a factory for the creation of new viruses. The enzymes encoded by the Paul gene are part of the pre-packaged mature virion that infected the cell and were released when the virus uncoded. As a reminder, these gene products include reverse transcriptase, integrase, and protease. And now that they're free inside the host cell, they get to work creating a new generation of viruses. First, HIV uses its reverse transcriptase to convert its single-stranded RNA genome into double-stranded DNA. And so our evil retroviral wizard is using his reverso transcriptome spellbook to turn his single-file RNA army into a double-file DNA army. Next comes integrase, another Paul gene product. This enzyme integrates the newly formed viral DNA into the host cell genome. Integrase will be represented by the keys the blacksmith produces to unlock the DNA tower so that the wizard's double-stranded DNA army can infiltrate. Integration into host DNA is a smart move on the wizard's part. For one, it can't be undone, and the host cell will be infected for the rest of its life. Plus, once the viral genome is incorporated into host DNA, The host cell machinery unwittingly transcribes and translates the viral genome alongside its own. Meanwhile, the ONV gene product, which is the precursor polyprotein GP160, gets glycosylated and then cleaved by host cell proteases into GP120 and GP41. This all happens in the endoplasmic reticulum and Golgi apparatus, because in eukaryotic cells, that's where membrane-associated proteins are processed. Remember, HIV is taking over the host cell's machinery, so it doesn't need to reinvent the process. Notice the reef around the ONV boat kind of looks like the ER in Golgi, the processing center for the virus surface proteins. You should know that while the ONV polyprotein GP160 is processed by the host cell, the GAG and POL polyproteins will be cleaved by HIV's own protease. So these polyproteins, along with other HIV components, make their way to the cell membrane. The immature virus then buds off the host cell membrane, forming the viral envelope from the host lipid bilayer along with GP120 and GP41. So we're done, right? Nope. Sorry. Remember, the POL and GAG gene products are still in their precursor polyprotein form. It's not until the end of the budding process, or shortly thereafter, that these polyproteins are finally cleaved by the viral protease, which is the third and last POL gene product. With all the products cleaved and in their active form, we've finally arrived at a mature virion ready to infect new host cells, and the life cycle is complete. So far, we've watched HIV mount a highly successful offensive. Of course, the host does try to fight back, though the key word here is try. Part of the host response is the formation of neutralizing antibodies. The primary target of such antibodies is GP120, the outer envelope glycoprotein. But the HIV wizard has performed a few magic tricks that hamper the host's antibody response. For one, GP120 displays a high degree of antigenic variation, which prevents the host from mounting an effective antibody response. In addition, dense glycosylation of GP120 shields its protein core from recognition by host immune cells and blocks the binding of neutralizing antibodies. As a result, the host is typically unable to form an effective neutralizing antibody response to the virus. Let's finish up by talking about treatment. All patients with HIV should be on antiretroviral therapy, or ART, to prevent the virus from wreaking havoc on their immune system. In general, combination therapy is preferable to monotherapy, in order to prevent drug resistance. Of course, it's no coincidence that we're bringing in pharmacologic therapy here in the life cycle sketch. These drugs have been designed to target specific parts of the life cycle. We'll start with reverse transcriptase inhibitors, NRTIs and NNRTIs. Let's break these down. NRTIs, also known as nucleoside or nucleotide reverse transcriptase inhibitors, are nucleotide analogs that competitively block reverse transcriptase. They may be the oldest drug class, but they're still the backbone of combination antiretroviral therapy. In the sketchy universe, NRTIs are represented by the noble knights of the Round Table. See how King Arthur is swinging around a hexagonal weapon? It's a nucleotide, right? Wrong! Don't worry, you're not the only one who's been fooled. To the unsuspecting eye, these drugs look like the building blocks of DNA. So these imposter nucleotides trick viral reverse transcriptase to incorporate them into DNA, and elongation comes to a halt. Take that, you evil wizard. NNRTIs, or non-nucleoside reverse transcriptase inhibitors, also halt elongation. But rather than binding the active site and being incorporated into the DNA chain, NNRTIs allosterically bind reverse transcriptase, altering its conformation and preventing further elongation. That's why Queen Elphavir, a non-knight, is trying to snatch the reverso transcriptum spellbook and stop the evil wizard. One important note, I haven't talked about HIV too much, but I should mention that it's intrinsically resistant to NNRTIs, so don't use those if your patient has HIV too. For more, check out our NRTI and NNRTI videos to learn more about how these drugs work and their side effects. Next are the integrase inhibitors, which, like their name suggests, prevent viral integrase from incorporating double-stranded viral DNA into host chromosomes. This is a very commonly used class of drugs, and for once, they're named in a helpful way. They all have Tegra in the name, like dolutegravir or raltegravir. Integrase inhibitors are generally well tolerated, but they're not completely without side effects, which is why this poor knight is looking a little beat up. Pay attention if your patient is complaining of sore muscles, as this could be a sign of muscle breakdown and inflammation, and check a CK level. Protease inhibitors target HIV during the last phase of the viral life cycle, maturation. Remember, the virus uses its own proteases to process the gag and Pol precursor polyproteins. And what better to represent the classic tale of a young boy's quest to mature into a knight than this young prince trying to pull at a sword, which is clearly stuck in the stone. You see, protease inhibitors kinda act like this stone, blocking final maturation of HIV virions by halting polyprotein processing. This renders these immature virions non-functional and unable to infect new host cells. Watch our protease inhibitors video to learn more about this class of drugs and find out if our friend ever gets that sword out of the stone. So far, we've talked about the drugs that target the major HIV enzymes, and therefore work inside the host cell or in the newly budded virion. But there are also agents designed to prevent HIV from entering the host cell in the first place. First up, Maraviroc, a CCR5 inhibitor. This drug works by blocking the CCR5 co-receptor on host cells, preventing the virus from gaining entry. Before the 120 siege tower can bind, in comes Maraviroc, a brave knight on a rearing mare. Maraviroc binds to the CCR5 receptor, so we've drawn the knight brandishing the CCR5 banner as well. Side effects of Maraviroc include rash, fever, vomiting, and rarely, hepatotoxicity. Since Maraviroc binds exclusively to CCR5, not the CXCR4 receptor, the viral tropism should be determined before starting therapy. There are also some newer entry inhibitors that directly block GP120, so they have a broader spectrum of activity, but they're reserved for special cases. If the virion had attached successfully, the next step would be membrane fusion. But before fusion can occur, in comes Infuvertide. Remember that fusion sounds like Infuvertide. This drug works by binding GP41, preventing it from undergoing the conformational change required for fusion of the viral envelope with the cell membrane. No fusion, no entry. Just like our cell membrane defender who's deflecting the invader's GP41 hook shots. Oh, also, as with NNRTIs, HIV has intrinsic resistance to Enfuvertide. Enfuvertide is given by injection, and it's likely to cause an injection site reaction. It can also cause other side effects like diarrhea, nausea, and fatigue. Wow, that grappling hook really did a number on that night shield. Yowza, that's gonna leave a mark. Okay, so what are you gonna do with all these antiretrovirals? Let's gaze deep into this crystal ball and find out. ART usually starts with a combination regimen consisting of 2 different NRTIs and an integrase inhibitor. So why do we mention all those other drugs? Well, this regimen is for treatment-naive patients, but patients may need to change therapies, whether that's because of resistance or side effects or some other reason. And that's when you'll get your protease inhibitors and other drugs involved. Alright, that's the end of our discussion of antiretrovirals, at least for now. But before we go, a cool genetics fact. You may remember from HIV Part 1 that CCR5 tropic HIV viruses dominate early on in the infection. The human genome has found a genius way to nip this in the bud. Individuals with homozygous mutations in CCR5, so-called supercontrollers, are virtually resistant to HIV and will never progress to AIDS. A naturally occurring Maraviroc, if you will. Well, that's all from us on HIV. Until we get a bunch of money to make a crappy prequel series with ugly CGI and a tacked-on love story. Not that I'm bitter or anything.